I'm wondering how many of you have tried to publish data generated with cell lines and got the reviewed paper back with a request for a more relevant cell system. I'm also wondering how many of you have considered primary cells for their project but were reluctant just because they were not familiar with primary cell culture. I can calm you down. Primary cell culture is almost as simple as cell line culture. It's just all about handling and best practice. I want to welcome everyone for today's webinar. My name is Claudia Schwartz, and I'm a scientific support specialist with Lonza. Thank you for joining us for today's webinar, where we will be discussing the why and how of primary cell culture. The presentation will take about 45 minutes, followed by a question-and-answer session. For this session, I have a good help. That's Dr. Susan Brecht from our scientific support team here in Cologne at Lonza. Hello, Susan.
Hello, good morning.
Okay, so if you want to submit questions to Susan, use the Questions tab on your screen. The presentation can only give general tips and tricks on primary cell culture. If you want to ask more specific questions, Susan will take care of these either at the end in the question and answer session or via email. There will be also at the end of the presentation a feedback form, and I would like to kindly ask you to fill that out as your feedback is quite important to us. We always seek to improve the quality of our webinars. Make sure that you don't submit your questions via the feedback form, as the form is not linked to your email address, and then your question is lost. A PDF copy of today's presentation will be sent by email to everyone who registered for this webinar, and you have also a chance to review the webinar on our archive on our website. If you have any technical problems, please click on the Help button, and our web host will assist you in resolving the issue. So, what are our topics today? I will start discussing the choice of cells to work with when starting a new research project. We will get more background information why cell lines are not the ideal cell model. I will then give tips and tricks on cell culture, handling of primary cells, and best practice. Contamination also plays a role in primary cell culture. We will talk about how to recognize and how to avoid contaminations. Finally, I will summarize the most important aspects of primary cell culture. Okay, so at the beginning of each research project, there is the question, which cells should I use for my study? Well, research, especially cell research, is expensive. So, hold on, this is your next slide. Okay, cell research is expensive, especially specialist equipment, training, reliable sources of cells, all that is expensive for research, and you need many cells. So that's why research is commonly conducted on cells derived from rodents or human material like cancer cell lines, immortalized cells, or in some cases you even have access to primary cells derived from section material. But these are all cell lines, and the reason for using these are they're readily available and they're cheap. So for initial research, there's nothing wrong about that approach. At least you get an idea about your theory, about what you plan to do could be performed in these cells. But how certain can you be that this cell line actually gives you the right result, that you're looking at the right effect? There are recently many concerns about cell lines. it has been reported, and that's just one citation where they actually talk about the use of cell lines in human research. They report about cellular cross-contamination, also about misidentified cell lines. So the cells you think you have, they're not the ones you have. And they talk about the use of cultures at high passage numbers. So the higher the passage number is, the more the cells went through genetic drift and are not the cells that they have been described as. And the number is quite concerning. This paper actually states that between 18 to 36% of all cell lines are considered to be contaminated with other cell lines or even misidentified. These concerns have been studied in more detail. So there are two studies I want to introduce here. Due to the better means to characterize cell lines, it has been revealed that the cell lines are not the cell type they have historically been reported to be. The first example, the study of Drexler et al., is a study about 500 reported human leukemia lymphoma cell lines. And the author of this study actually states that 15% of these were found to have been misidentified. So they're not the cells you think they are. The second study I want to highlight here is the study of Sean. They studied the HEC293 cell. These have been generated by adenovirus transformation of human embryonic kidney cells. So they are supposed to be some kind of kidney cell, but in this study they actually could show that these cells have many properties of immature neurons. So the theory now is that the adenovirus was taken up by a neuronal lineage cell in the original kidney culture and not a kidney cell. So think about using the HEC293, which is a frequent in vitro model for kidney cell function. You study kidney, and what you work on is immature neurons. So think about all the data you have generated with cell lines over the years. Isn't it somehow concerning that you need to question the validity of the data? That is, it really matters whether you use primary cells or cell lines. I want to show with the next two slides. So in the first slide, an experiment is shown that is performed on human T cells compared to a human T cell line, the Jurkat cell line. And an siRNA experiment has been performed using an siRNA directed against vimentin. Vimentin is an intermediate filament protein part of the cytoskeleton. And you see that the downregulation of vimentin messenger RNA determined 24 hours post-nucleosection shows that the downregulation in human T cells is 70%, the downregulation in Jurkat cells is 80%. This difference is significant, as you can see from the arrow bars. You may now think that the impact of that difference is of minor importance, but have a look at the next example. This is a study we have done on HEC293 cells. We compared the effect of camptothecin on primary kidney cells and compared that with the HEC293 cells, the in vitro model for kidney cell function. And we studied the impact of camptothecin, the cytotoxic drug. Camptothecin derivatives of camptothecin derivatives actually are currently investigated as potential chemotherapeutic therapies for a number of cancers. So there is an impact of the behavior of HEC293 versus primary kidney cells. So what we've done is we've done a dose-response experiment, and we measured the viable cells after increasing amounts of camptothecin. And we measured this using our ViaLight Plus assay, an assay based on the luminescence measuring the ATP donated to the assay reaction by viable cells. And you see that the dose-response curves are quite differing. While you need about 70 nanomolar to kill 50% of the HEC293 cells, you need almost tenfold of that concentration to get the 50% killing in primary cells. So I hope now that you're convinced that not cell lines, but primary cells are the only model system that gives reliable results. What are then the advantages of using primary cells from Lonza? As we use non-immortalized and non-transformed cells, they look and act like normal cells. We intensively QC these cells, and you can be sure that you work with the cells that you purchased from us. Also, it's quite convenient just to take the vial out of the liquid nitrogen. You don't have failed isolations. You can start your research right away. You don't spend time analyzing or building up tools, but you can start right away to analyze your results. Another advantage is the ease of use. So we don't only sell the cells, but we also sell the media that comes with growth factors, so you don't have to do any research, any studies on what media you want to use for your primary cell. We also offer a protocol and technical data. So the culture of the primary cell from Lonza, together with our media and our subculture reagents, are tested together. They are guaranteed with the protocol, and it's quite straightforward. And what you do is you actually avoid issues with inconsistent supply. So whenever you need cells, you can get them. You get qualified and tested and QC'd cells. And the lot-to-lot consistency actually is great with our cells and with our media. So these are the advantages of using our cells. Now, knowing that cells are the only reliable system for cell research, why do so many researchers still stick to cell lines? First of all, they have concerns about using primary cells for their experiments, and they are not familiar with primary cell culture. What are then the concerns people have? Well, the feedback we get from our customers is that they fear low proliferation rates, they fear bad reproducibility, they fear that low functionality could hamper their research, and they are not sure about a proper freeze and thaw technique. Also, viability issues after thawing of or passaging of cells are concerns people told us, or low viability after detachment of the cells. There could be also issues with functionality and differentiation. And last but not least, you could have problems with contamination during the isolation procedure. So these concerns actually can be circumvented using our complete cell system. So we offer a combination primary cell, proliferation, or differentiation media system in bullet kit format, and the reagent pack subculture reagent. And by combining these three items, there is no risk of actually failing the culture. But still, using good quality cells plus optimized media systems and cell culture protocol can still cause problems. There is no guarantee. And guess why? Well, there's still you as researcher who needs to know how to handle the cells. You need to have a good best practice to do the handling properly. And therefore, I want to go stepwise now through the handling process of the primary cells, and you will see that's quite straightforward, and it's not really different from cell line cell culture. The only thing is preparation is key. So here an outline of the thawing procedure. And I quickly want to show you how important the steps are, although they're probably easy to understand. I just had a customer calling in for a problem with viability of bronchial epithelial cells. They actually called after they thought the fourth vial of cells with low viability, or even almost just dead cells. So a brief troubleshooting revealed that they thought the cells with gloved hands. And gloved hands means no temperature at 37 degrees, and the temperature transmission to the cell is far slower than a water bath. So here, make sure that you use the best practice we introduce here in the webinar or in our protocols to make sure you avoid these kind of issues. So sticking to the protocol is crucial. So preparation is first. That's a key point. So prepare the flasks with media containing all necessary growth factors and put them in the incubator to get them equilibrated to the right temperature, 37 degrees, and to the right pH. Also prepare the right number of flasks. You need to know in advance how many cells you have in your cryovial, and then you need to calculate the volume you want to seed in these flasks to reach the ideal seeding density. That's also very important to have that calculated beforehand. If working with our clonetic cells, that's easy. We give a certificate of analysis that gives you the cell number that is actually in your vial. And we have the protocol, and this gives you the ideal seeding density of the cells. So once you have that prepared, take the cryovial from the liquid nitrogen where it's kept as long as possible. So never take the cryovials out beforehand. So then, once the cells are out, you start the thawing procedure. Put the cells in, or hold the cells in liquid, in water at 37 degrees in a water bath, and to thaw the cells quickly. And this needs to take less than 2 minutes. Everything above that is harmful to the cells. And then there is one point, don't spin the cells at this point. You do remove the DMSO at a later point. We performed quite a few studies that spinning is much more harmful than leaving the DMSO for the first 24 hours in the cell culture. So now that the cells are in the medium, now that the cells are in the medium, you need to do the first medium change after 24 hours. And actually, you do this medium change to get rid of the DMSO and the dead cells. Then you do a medium change every 40-80 hours until subculture. Make sure that you don't do a repeated warming and cooling of the medium, as it contains growth factors and supplements. They are temperature sensitive, and the more you heat them, the more they actually are not working anymore. So prepare aliquots of the medium and just take the aliquot to warm this to feed your cells. So the seeding density at day 1 is, well, there are some cells perfused under the microscope. Don't expect the cells confluent at day 1. So they start growing once they are attached and once you did the first medium change, and they catch up to get the normal proliferation rate, which is around one population doubling per 20 hours. And when the cells reach a confluency of 50%, make sure that you increase the medium volume, because otherwise you could give the cells a little starving when not adding enough medium. The cells should be subcultured at a confluency of 70 to 80%, and that's usually reached between 5 to 9 days. Make sure that you don't reach a 100% confluency. At this confluency, the cells get contact inhibited, and they could go into senescent. And even if the cells are processed afterwards and they may catch up with proliferation, it's not sure whether they return to the normal phenotype. Okay, now that the cells reach the 70 to 80% confluency, they need to be passaged. So for subculturing, we recommend the reagent pack from Lonza. For clonatic cells, it's a must. Otherwise, you lose the warranty. And there are several reasons why we actually recommend the reagent pack. First of all, trypsin concentration is crucial. Primary cells are especially sensitive to trypsin, and therefore in the reagent pack there is a trypsin which has the ideal concentration for primary cells. Secondly, the stop solution to neutralize the trypsin is also crucial. In cell line culture, you're used to take just media, but for primary cell media, you have sometimes media that does not contain enough reagents that neutralize FCS or even no neutralization agents like FCS, for example. So in the TNS, there is enough protein to actually inactivate the trypsin. So in general, the trypsinization process is just like for cell lines. The only thing, again, is preparation is key. So make sure that you have the flask with media in the incubator to get the right pH, to get the right temperature, and all reagents for trypsinization need to be at room temperature. So, and when we start the subculturing, we actually start removing the media and wash the cells with washing buffer, which is HEPAS buffered salt solution in the reagent pack. Then when taking the washing buffer away, we add trypsin, and here it's crucial to do this at room temperature and track the trypsinization process under the microscope. Once the cells round up, do a firm wrapping on your palm that detaches the cells from the culture vessel. Once that's done, you need to immediately neutralize the trypsin with TNS and not medium, as I mentioned already. Now you spin down the cell to remove the trypsin, and the cell pellet needs to be resuspended with a small volume of media, and then you place the cells in the flask at the ideal concentration. So you need to do a cell count before seeding the cells in the flasks. There are other methods than trypsinization to detach cells from the culture vessel. There is trypsin or acutase treatment. These are trypsin alternatives considered less harsh for the cells. There's also cold PBS that only works for loosely adherent cells, and there's scraping, but this is only for really few cell types. So what do you take into consideration when working with clonatic cells? First of all, there is the donor variation. So it depends on your purpose, what you want to study, whether you use the cells from the same lot or from different lots. if you want to keep the variance in your assay as low as possible, make sure that you work with cells from the same lot. You can reserve our cells that you have from the beginning, the same lot available for your complete study. Different lots would be useful if you want to examine how much a pathway or an effect of interest vary across the population. Then there is the freezing and thawing of the cells. It is stressful. And even if you seed the cells at the recommended seeding density of, let's say, about 5,000 cells per square centimeter, it looks like very few cells in the microscope. But that's normal. Just aspirate off the media and the floating cells, replace with fresh media, and the remaining cells will proliferate and should be confluent in 5 to 10 days. Then there's one important aspect of the cells, of every primary cell, not only clonatic cells. Primary cells have a finite lifespan. The overall lifespan is more or less 50 population doublings. But this is far too close to the phase where they go into senescence already. So we actually guarantee our cells for only 10 to 15 population doublings. After these, they actually come closer to the end of their lifespan, and the morphology and the cell performance will begin to change, and the growth will slow down dramatically. So now, what about the growth factors and supplements in the media? When do I need growth factors? Well, you only need growth factors when you want to keep the cells alive for a time frame more than 24 hours. And what supplements are important? Well, of course, we give the supplements to our media because we know adding these to the media, the cells are completely happy. But do you need them all? Well, that depends on your application. You can, for example, for a certain experiment, leave out one or another factor. For example, endothelial cells still proliferate okay for 1 to 2 passages without the hydrocortisone. But this is cell type specific, so you need to find that out for every cell type you want to study. And if you want to differentiate your cells, we also offer bullet kits that provide differentiation to the cells. For cell types like human mesenchymal stem cells, pre-adipocytes, normal human articulate chondrocytes, and normal human osteoblasts, we do offer differentiation media. Now that the cell culture works fine, there's nothing else that can go wrong. Well, unfortunately, not really. There is still something that can go wrong. For example, the contamination. So, in cell line cultures, contamination are limited due to the addition of antibiotics. What about primary cell culture? Do you use also antibiotics in primary cell culture? Well, we know that high levels of antibiotics can be detrimental to primary cells by stunting growth or decreasing viability. For our bullet kits from Lonza, you don't need to add extra antibiotics as we add gentamicin and amphotericin B to our bullet kits, and they are actually at an optimal concentration to prevent low levels of contamination, but still maintaining cell health and viability. The gentamicin would be a general antibiotic for a huge variety of different germs, and the amphotericin is an antifungal agent. So the good thing about most contaminations is you can see them by eye. So whenever you look through the microscope, check for contaminations, whether you see something that you can see which doesn't belong into your culture. There is, for example, black specks These are the bacteria, so they can be easily noticed. They also induce a pH change, a drop of pH, which leads to the media turning yellow. But be careful, they can also be mistaken for cell debris. So whenever a cell dies, you have cell debris on the cell culture surface, so that's not contamination. But the other way around is something we actually learn much more often from customers is taking place. So a lot of debris or precipitates in the medium are mistaken for contamination, especially those customers working with epithelial cells or endothelial cells. They have to add bovine pituitary extract or bovine brain extract or serum into the media, and that can form protein precipitates, and they are quite often mistaken for bacteria. But make sure you have a close look. The precipitates, they do a Brownian movement, which has nothing to do with the directed motion or the definite movement of bacteria. So have a close look, and you can distinguish between bacteria or precipitates. Then there is the possibility of getting a fungus in your culture. They are easy to identify. They form filamentous strands, and at a later stage, they even form web-like meshes. So, sorry, I forgot to send that slide. So here the bacteria as black spots, and here the meshes of the fungi. Okay, so then I actually move to the next slide. So another germ that can contaminate our cell culture are the yeast. They are also easy to detect in the microscope. They are of oval and round shape. They are smaller than cells, and as they reflect light, they appear as bright beads in the cell culture, easily seen here on the slide. And the more advanced your contamination is, the more they form branch chains. And, well, last but not least, they are mycoplasma. Unfortunately, these are not visible in the microscope. You just notice that your cells are not acting quite right. And let me tell you a short story, what I experienced as a PhD student. I worked on a DNA-binding protein, and I studied that protein in a so-called electrophoretic mobility shift assay. I incubated my nuclear protein with radioactively labeled DNA sequence, and I separated that through a gel. And all of a sudden, I detected a new protein that bound to my DNA sequence. I was so enthusiastic that I discovered a new DNA binding activity. But then, guess what? That was just mycoplasma related. So mycoplasma induced the DNA binding activity, and once we threw the cells away, there was no new protein. So mycoplasma do impair a lot of functionalities of the cells. That's another example that we got from a customer. HeLa cells transfected with a nuclear factor technology with a GFP expression construct. And what they routinely got with normal HeLa cells, healthy HeLa cells, was about 60% GFP expression analyzed by FACS. In the infected cell line culture of HeLas, they just obtained 23%, so there was a drop of more than 50% in transfection efficiency. So that also is affected, transfection efficiency is also affected by mycoplasma contamination. It has been quoted that mycoplasma infections affect every known process in the cell. I just want to highlight a few of them. So the effects of cell growth, cell metabolism, could be affected. There could be disruption of nucleic acid synthesis, and even chromosomal aberrations can occur in cells when infected with mycoplasma. There are actually studies that quote that 10 to 20% of all cell lines are infected with mycoplasma. You'd probably agree that mycoplasma infection is a major problem of cell culture. So what are these mycoplasma that you can't see in the microscope and are so powerful? Well, they're the smallest, simplest prokaryotes of a size of 0.2 to 0.8 micrometer. They can't even, or many of them, can't even be removed by filtration. They even pass a filter of 0.2 micron size. So due to the lack of a rigid cell wall, they're not affected by traditional antibiotics used in cell culture. And they use the nutrients from host because of their limited biosynthetic capabilities. Where do the mycoplasma contamination arise from? Well, there are several possibilities, but most common are the following two. There is you as manipulator. The majority among us is a carrier of mycoplasma in our mouth. So each time when you sneeze, cough, or even talk in front of the hood, you could contaminate your cell culture already with mycoplasma. The other quite frequent possibility of infection is getting cells from other sources. And these cells are actually cultured in the same hood, in the same incubator, and the same media, having the mycoplasma infection. And then the mycoplasma are easily transferred into other cell culture vessels. So to avoid contamination by best practice is the only goal you can have. What does best practice mean for primary cells or cells in general? Once you receive new cells, put them in quarantine and test for mycoplasma. Quarantine means ideally a separate incubator, but if that is not available, a plastic lunchbox with some damp tissue in it is also sufficient. When the mycoplasma testing is negative, okay, you put the cells in normal culture, you prepare your cryo samples, you actually can use these cells for your experiments, but still monitor the cells on a regular basis. That's part of best practice. So what happens if the cells are positive? You can keep the cells in quarantine, treat them with Mycozap, our antibiotic mixture that actually kills the cells, inform the source. That's just fair to do that, and monitor the cells daily for mycoplasma titer, and keep the cells in quarantine until the infection is eradicated. But the best way to actually handle mycoplasma contaminated cell culture is just destroy the cells. As you don't know if the clean cells ever truly recover from all cell functions, you don't really know what to work with, and that's the only option if you have precious cells. Any other cell culture just should be thrown away, and again inform the source where you received the cells from. There are many options how to test for mycoplasma. The MycoAlert assay from Lonza is a very fast, a 30-minute assay that gives reliable and easy-to-interpret results. And that's compared to PCR amplification, fluorescent staining, and the culture method. The MycoAlert assay is based on the luminescence reaction and a mycoplasma-specific enzyme involved in ATP synthesis. What happens actually in that assay? here a schematic of the MicroAlert Plus assay. So on the left side, you have your reaction vessel, and you start with the sample. This is the cleared cell culture supernatant of your cells. Then you add the MicroAlert Plus reagent that contains the lysis reagent, the luciferase, and the luciferin, and you incubate for 5 minutes. What happens in these 5 minutes? Well, if there are mycoplasma present, they get lysed, and they release a mycoplasma-specific enzyme that is an enzyme involved in energy metabolism, so is able to generate ATP. So then there is luciferase and luciferin in the reaction tube already. So whenever there is ATP in the sample from FCS, for example, that generates light in the luciferin-oxyluciferin reaction, and that's measured in the reading A. So after 5 minutes, a first reading is performed, and that gives you the background light of your sample. And then you add the MicroAlert Plus substrate. So that contains the specific substrate for the mycoplasma enzyme. So whenever that enzyme is present, the substrate is turned into ATP, and that actually helps the luciferin to turn into oxyluciferin and to generate light. And then you incubate that for another 10 minutes and do the reading B. And how to actually calculate the result? That's really easy. You do a B to A ratio, and if the ratio is below 1, the sample is negative. If the ratio is above 1.2, the sample is positive. And if the ratio is between both numbers, put them in quarantine and check in like 3 days, for example. And if the ratio stays the same, it's just background, and you can actually take the cells in the normal cell culture. In case of a mycoplasma contamination, you need to have an increase in the ratio. So here a few details on the MicroZap reagent. It's a reliable method for mycoplasma elimination, and it has minimal toxic effects on cells. It's a combination of antibiotic and anti-metabolic agents. So that's all I wanted to say about the MicroZap here. If you have more questions on that, feel free to ask these. So now that I nearly reached the end of the presentation, let me briefly summarize the key points. Well, cell lines are often unreliable and unauthenticated, leading to inconsistent data. Lonza primary cells are the superior model for research due to documented sources and strict QC. And the most important point, probably, cell handling is crucial for success. Pay attention to culture conditions and follow the specific protocol. Contamination is a major contributor to cell culture problems. Be sure to check your culture regularly. Lonza primary cells and media are tested to guarantee sterility and mycoplasma-free products. Before we move on to the question and answer part, I want to introduce our online databases. So we do have a cell database online that gives information on cells, the media to be used with these cells, and how the cells are characterized. Then we have a media database online where you can check the media, their characteristics, and the shelf life of the media. And we do have a citation database that consists of more than 2,400 peer-reviewed papers. And you can actually use the comprehensive search engine to look for papers where Lonza products have been used. If you want to talk to our primary cell culture experts, you can use the email address displayed here. So it's the scientificsupport.eu@lanza.com, or you can give us a call and we are happy to talk to you directly. So I would now briefly remind you that we have the feedback questionnaire at the end, and I would like you to fill that out and make sure that you don't use this form to question. Please still use the feedback tab or the question tab, on your screen. So while you have a few seconds to actually go through that feedback form, I want to use that time to go through our questions and prepare them for the question and answer part, which is following the form. Okay, I think probably most of you are through the feedback form. Susan, are you ready for getting questions?
Yes, I'm here. Hello, yes.
Okay, so we got quite a few interesting questions, basically. And there's one question which is, I think, quite important, and therefore I want to mention it first. There's a customer who wants to get a specification or a more specific, the difference between population doubling and passage. Can you comment on that?
Yes, well, this is a very important difference. One population doubling is really the time or when the cells double in number, while a passage is the removal or the subculturing of a cell population from a vessel to one or several other vessels. So more or less a trypsinization. It's very important to see here the difference, because for many of our clonetic cells, we, for example, guarantee 10 to 15 population doublings. However, if as one population doubling, as within one passage you have more than one population doubling, usually you have 3 to 5 population doublings, these 15 population doublings are equal to approximately 5 passages after thawing.
Okay, I think that's clear now. Then there is another question. A customer using HUVEC cells wants to refreeze the cells after he doesn't specify the number of passages, but is that in principle possible with our cells?
In principle, it is possible to refreeze our cells, but please be aware that we cannot guarantee the performance of the cells after cryopreservation. Why? Simply because cryopreservation is a very complex procedure. But I have to say that many of our customers are successfully refreezing our cells, and we also have recommended protocols for refreezing them. There are only some exceptions for the cells which can't be refrozen. This is mainly cells which are non-proliferating, like neuronal cells, hepatocytes, osteoclast precursors, for example.
Okay, then here a question. Do I need to heat inactivate the serum, so the FBS contained in the single quad kit?
No, so the serum contained in the single quad kits is not heat inactivated, and we also do not recommend to heat inactivate the FBS. Heat inactivation can denature essential components in the serum, like growth factors, vitamins, etc. And if you heat inactivate the serum in the single quads, then usually you will see that the cells will not perform as good anymore.
Okay. So then a question that probably is a question of more than one customer. We don't have a liquid nitrogen tank. Can I store the cells at -80?
No, this is also something we do not recommend. Primary cells should really stored immediately upon arrival in liquid nitrogen or in vapor phase nitrogen. To store the cells at lower temperature, at higher temperature, for example at -80 degree, or in these ultra cold freezers which have -120 degree, is not recommended.
Mm-hmm. Okay. So then there's a customer who's writing, I'm not familiar with primary cell culture. Do I need to coat the plate for a special, for example, with fibronectin or collagen before plating primary cells? They don't specify actually which cell line they're interested in.
Well, okay, for most clonetic cells, for many primary cells, if you're using our medium and our clonetic cells, it's not necessary to coat the plates. There are for sure some exceptions, but if you have to coat the plates, we are mentioning it in our protocols, and we also recommend which coating to use and at which concentration to use the coating.
Mm-hmm. Okay. And then there is a question on FCA or human serum albumin. So do you recommend to use FCS or human serum albumin for primary cells from human origin?
I have to say this depends on the customer's preference. In our clonetics media, still most media do contain either FCS as undefined supplement or BPE as undefined supplement. However, we are also we are now also offering some media which are chemically defined, which mean they do not contain this undefined supplement, and often in this case they might contain HSA or other proteins. But it's not possible to simply replace FCS by human serum albumin, because for sure in FCS you have many growth factors, many vitamins, whereas human serum albumin is just a protein, so it adds no growth factors or anything else to the culture.
Okay. So there is an interesting question on the antibiotics used in EGM2. I use your EGM2 media for culturing endothelial progenitor cells. Is there a reason why you shouldn't use the standard PenStrep instead of gentamicin with endothelial cells? Do you have an answer to that?
Well, in our clonetics media, we decided to choose the gentamicin for two reasons. First, the gentamicin is not as toxic as penicillin, so the difference between effective concentration and toxic concentration is bigger for gentamicin. Also, PenStrep is in use since the early days of cell culture, and so many resistant bacteria have developed meanwhile, while for gentamicin there are less resistant clones. And as mentioned, we would not recommend to replace the gentamicin in our clonetics media by PenStrep. There is really no advantage to do so, and if you choose too high concentrations, the cell might even suffer.
Okay. There is a question whether we provide a general media for primary cells of any type, for example, for cells with unknown origin.
Well, it's very hard to make a general medium, especially if the cells are of unknown origin, because the cells have different requirements. So an epithelial cell has very different media requirements with regards to an endothelial cell, for example. So our Clonetics media are really all specialized for one cell type, like for endothelial cell, epithelial cell, or similar. We are also offering some serum-free medium, which we call, and there in our serum-free media range, we are also offering media which we call general purpose serum-free media. That these media might work for some primary cells, but not for all of them. So you can try such a media, but usually it's very hard. At least you have to know at least the cell type. So if you have endothelial cell or epithelial cell or fibroblast, to choose a medium specialized for this cell type.
Okay. Then there is a question again. There's quite a few questions on antibiotics. Can we add 5 times antibiotics initially for primary cultures and then shift to 1-time concentration?
So if you're using our clonetic cells, which are isolated cells and which have been tested for sterility, the answer is no, you should not do this, as this can kill the cells. I suppose this question is more going in the direction if a person is isolating cells himself. What you then often do in those cases is that at the beginning you are washing the tissue, so not the isolated cells, but the whole piece of tissue with an increased concentration of antibiotics. There you can go a bit up with the concentration of antibiotics to really kill the bacteria which are attached to the tissue. However, once you have really an isolated cell, I would not recommend anymore to use such high antibiotic concentration. Anyway, to use a 5-time concentration of an antibiotic is really very high, because for some antibiotics you will already go from the effective range to the cytotoxic range. So it also depends a bit on the antibiotic. There are tables, for example, which show effective and toxic concentrations for an antibiotic. And to be true, it makes not too much sense to increase over the effective dose of an antibiotic because, if this is the effective dose, it will kill the bacteria. To add 5 times more, it will not kill 5 times more.
Okay, so I would say we are almost we are through all the questions. There are just a few very specific questions. We will actually get back to you by email, and I would like to thank you, Susan, for your answers. That was really helpful to get your answers here in this question and answer session. I would like to thank you all for joining our webinar today on primary cell culture. And I hope you enjoyed it, and we are happy to see you again for our next webinar. Have a look at our website where we actually advertise the next upcoming webinars for the rest of the year. Thank you very much and have a good day.